Well, welcome everybody. My name's Stephen Robertson and I'm a clinical and molecular geneticist, and I'd like to extend a warm welcome to all of you for this special public lecture, guest lecture, as part of the GSA conference. It gives me great pleasure to be able to introduce one of Australia's own, a world leader in his field, to address us tonight in a very rapidly and exciting evolving area of human genetics. Daniel MacArthur has multiple appointments in Boston at the Broad Institute, MIT, Harvard and the Mass General Hospital. He's made his name in establishing and then analysing very large and ever-expanding data sets of human sequence data to further understanding about human genetic disease. Daniel completed his PhD in Sydney before moving to Cambridge for his first postdoc at the Sanger Centre at Hingston in Cambridgeshire, before moving to the Broad Institute in Boston, where he currently resides. So it's with great pleasure I introduce Daniel. I think we're in for a treat, and there'll be an opportunity for a bit of Q&A afterwards. Daniel.
Thank you very much, Stephen. Can people hear me at the back okay? Or is that too loud? Feels a bit too loud, perhaps? Not too loud? Okay, all right, thank you very much. All right, well, thank you so much to Stephen for that very kind introduction, and of course to the organisers for the chance to come out. It is always a delight to be back in the Southern Hemisphere, even in winter. So as Stephen said, my group is pretty focused on different ways of taking very large-scale databases of human genetic variation and using those to improve our interpretation of the types of genetic changes that we find in patients with rare disease. And what that means in practice is that my group basically spends about half its time thinking about biology, and in particular, the biology underlying specific rare diseases like muscular dystrophy. And the other 50% of our time we actually spend thinking about things like software engineering and data sciences. And a lot of the work that I'll be presenting today is really a kind of fusion between those two different areas, thinking about how we can leverage the just enormous amounts of data that are now being generated in the context of human genetics and turn those into things that are actually useful to us for interpreting human genetic variation, but that also become useful for the rest of the community as well. And so in the talk this evening, I wanted to start by, I guess, describing why I think this particular point of time in human genetics is an absolutely amazing time to be doing human genetics. I would argue the best time in the history of the field. I'll then go through a relatively brief description of two of the data sets that we've been working on now, I guess, for the last 4 years, the EXAC and Nomad data sets, very large collections of human genetic variation. And I'll talk a little bit about how we built those and some of the politics behind them, and then also how we can use them to understand more about human genes. And then I wanted to tell two stories, one about a thing we call the Human Knockout Project, which is an exercise in trying to identify very rare individuals who are completely lacking the function of particular human genes, the human equivalents of knockout mice, and using them to understand more about genes that where we don't fully know the biology yet. And then the final story, which I'll save to the end, is a story, actually a pretty personal story about the ways in which a person with a very strong incentive to learn more about a specific disease can leverage these large collections of human variation to understand more about how individual genetic variants actually influence the risk of disease. So let me start by talking about why human genetics is so amazing at this particular point in time. And I like to think of human genetics as important in part because it provides us with a lever for moving between two very different kinds of information that we as a community have become extremely good at generating. And the first of those kinds of information, which is encapsulated in this relatively random screenshot from the UCSC genome browser, which I suspect many people in the room are familiar with. But basically what this browser allows us to do is to look at a section of the human genome, some chunk of the 3 billion bases of the human genome, and learn more about what each of those bases does. So if that base is found within a gene, it will tell us that it is present in a gene. We can look up which tissues that particular gene is expressed in. If it's found outside genes, we can learn more about whether that particular region is involved in the regulation of gene expression and across what tissues. And as efforts like the ENCODE project and epigenomics roadmap have gotten up and running, we can learn more and more about the functional layer, this layer on this functional annotation on each of the different points in the human genome. A second type of information that we have been developing really, I guess, over many decades is information about human phenotypes. That is the collection of all of the different pieces of information that make up how a person appears and behaves and feels and the types of diseases that they have. So national biobanks now contain many hundreds of thousands, in fact millions of humans. And for those people, we often have information about all sorts of things. So we can look up their disease diagnoses in electronic medical records. They have information about prescription registers, what drugs they're taking, whether or not they have had cancer at particular points in time. Increasingly, as we develop functional genomic technologies like metabolomics and transcriptomics, we can look at that data as well for all of these individuals. And that means we can really start to build up a picture of the biology of those humans, those individual humans. And then the nice thing about human genetics is it allows us with ways of moving between these two worlds. And we can do this, we can actually move in both directions here. So in the context of standard human genetics, what we would typically call forward human genetics, what we tend to do is to take people who have a particular disease. So this might be a set of families with a specific rare disease like muscular dystrophy, or it might be a set of families with a set of individuals with a common complex disease like type 2 diabetes or rheumatoid arthritis. We take those people and we look at their DNA using one technology or another, and we can use that to then move towards particular genes that are implicated in that specific disease. And that approach, this forward genetics approach in humans, has been extremely successful. It has allowed us to identify thousands, nearly 4,000 genes now associated with rare diseases. We've also identified many thousands of regions of the genome that are associated with common complex diseases through genome-wide association studies. And so that approach really has allowed us to move from human phenotypes right the way through to some understanding of the underlying biology. Now, as our data sets of human genetics get bigger, it becomes feasible to think about moving in the opposite direction. That is to start with individual genes, to find people who have genetic changes that perturb the expression or the function of those particular genes, and then collect those individuals and learn more about what their phenotype is. This is called reverse genetics. Of course, this approach is well established in the context of model organisms, where we have the luxury of being able to actually directly interfere in an organism's genome and make changes to a particular gene. In the context of humans, we can't do direct genetic engineering for a combination of logistical and ethical reasons, but what we can do is take advantage of naturally occurring human variation to find exactly the people who actually have changes in a particular gene, and then again collect them and learn more about their function. And the reason this is so powerful is because there is so much variation that exists in the human population. So there are more than 7 billion humans now currently alive. Every single one of us carries something like somewhere between 60 and 80 mutations that are present in us, but were not present in our parents. These are de novo mutations. So if you add up those numbers, 7 billion humans and 60 to 80 new mutations, and only 3 billion bases that exist in the human genome, it's actually very clear that the vast majority, in fact, probably all of the single base changes that can exist in the human genome that are compatible with life do exist somewhere out there in the human population. So somewhere out there is at least one, and often hundreds or thousands of people who carry any specific base change that you could care to be interested in engineering into the human genome. So that means that basically the human population consists of a whole collection of individuals with very specific genetic perturbations. Each of us effectively is an experiment of nature, and all we have to do is find those individuals who carry that particular change in a specific gene and look at those individuals to learn more about what that gene does. So that, of course, is slightly ridiculous when we're talking about 7 billion people, most of whom we don't actually have sequenced. But it starts to become a little bit more feasible as we start to generate the scale of data that I'll be talking about in a couple of slides' time. So before I get to that, I just wanted to emphasize some of the pros and cons of thinking about humans as model organisms. And I know there's many people here who work on various other model organisms, many of which are much simpler to deal with than humans. So I thought it was worth actually articulating some of the challenges and the benefits of humans in this space. So the challenges include the fact that humans have a very long breeding cycle. They have disappointingly few offspring compared to mice, or certainly compared to worms or flies. And also they have this terrible habit of random mating, or at least random with respect to what geneticists want them to do. So as a result, we can't set up the population in the way that we would in the context of a particular model organism. Although, as I'll show later on in the talk, there are particular types of population that we can look at that make it more likely that we will find out the model organisms that we're going after. A second big problem with humans is that we can't engineer their genomes, and that is a problem in terms of generating particular types of alleles that obviously are never going to exist in nature, so things like conditional knockouts. Nature is not kind enough to ever give us a mutation that switches off a particular gene in particular tissues or at particular points in time. So we don't have that flexibility that we have in model systems. There are major limits to interventional studies for many ethical reasons. There are certain types of experiments that just can't be done, thankfully, in humans. And they're incredibly expensive to house and feed, although many of them do actually manage to pay for themselves, so that makes life a little bit easier. So those are some of the challenges, but there are also considerable benefits to using humans in this setting. The first is that their biologically is unambiguously relevant to humans. We don't have, when we're making an argument in our grants about the fact that the biology we study is relevant to the human situation, that's much easier than if we're studying mice. And I should say I did my PhD studying mice, so that makes our life a lot easier. There are enormously large numbers of humans available that we can actually study. There are very well-established systems for phenotyping them. They're called medicine. And we also have these large, well-established phenotyping centers called hospitals that collect long-term observational data from these organisms over a long period of time. And although it's often not collected in the perfect way for research studies, it does mean that we can actually get, if we have enough sequenced individuals, we do have lots and lots of data that we could theoretically use to connect to human genetics to learn more about the functions of genes. And we are now starting to live in a world where the amount of sequenced humans is growing absolutely exponentially, and I mean that literally exponentially. So it is the case that now around the world, over 2 million humans have had sequencing done on a genome-wide scale. And by genome-wide, I typically mean one of two different technologies has been applied. Either whole genome sequencing, which looks at all of the bases, roughly all of the 3 billion bases in the human genome, or more commonly exome sequencing, and you'll hear that term come up a little bit later on. For those of you who don't know what an exome is a very cost-effective way of looking at the roughly 2% of the human genome that codes for proteins. So that's the bit of the genome that we understand the best, and it's also the bit of the genome where most of the changes that underlie rare disease are found. So exome sequencing is a really good way of looking at that. So we have over 2 million people who've now been sequenced, and they've been sequenced in a whole range of different settings, from large-scale academic sequencing centers right the way through to big medical centers, as well as a bunch of projects currently underway that are being financed by pharmaceutical companies as well. Just to give you a sense about how rapidly the scale of sequencing has gone up, these are numbers from just one place in the world. This is the place that I work, the Broad Institute of Harvard and MIT. The Broad has the largest genome sequencing facility in the US. And over this, what this plot shows, for those of you who can't read the numbers, is from 2009 up to the end of 2016, how many genomes we've sequenced on the left-hand side and how many exomes we've sequenced on the right-hand side. And the numbers, for those of you who can't read, are we currently have in our hands just over 70,000. This number is a little bit out of date. Just over 70,000 whole human genomes that have been sequenced, and somewhere north of 250,000 exomes that have been sequenced just in one place in the world. And so those numbers are impressive, but also the growth of these numbers is perhaps the most important thing, and that's particularly true for whole genome sequencing. As that technology has begun to explode, we are now generating more data in any given year, typically than we have generated in all previous years combined. So this data the amount of data is increasing at a truly ferocious rate. And this is really a lot of data, and I don't just mean that by biology standards, I mean that by anyone's standards. So one genome, the amount of data that goes into one genome is about 120 gigabytes of information. The average Netflix user, at least in the US, I have no idea about Australia or New Zealand, the average Netflix user streams about 45 gigabytes of data per month. So basically it would take you about 3 months to stream enough data through Netflix under average consumption to get approximately the same amount of information that you have existing in your genome in all the cells in your body. The Broad currently sequences about one human genome every 10 minutes. So it obviously takes a lot longer than 10 minutes to sequence a human genome, but that's how often they roll off the machines. And that means that we build up the equivalent of the entire Netflix library every 2 months. There's a lot of crap in the entire Netflix library, but most of this information is actually pretty valuable. And in total, in the Broad data center, we have about 10 petabytes of data. And that is a truly staggering amount of data. So to put that in perspective, if you were to take all 10 billion photos that exist on the Facebook servers, that only takes up 1.5 petabytes. So that's about 1/7 of the total amount of data that we have on our servers. So here we are talking about, so biology, at least human genomics, is very rapidly becoming a data science and involves a scale that is truly impressive, not just to us, but to other people as well. And I should say, actually, one of the things we like to brag about at the Broad is that twice over the last year, we have managed to break the Google cloud. which is actually pretty hard. Google is very good at engineering clouds that are robust, but we've been generating and computing data at a scale that has actually made it very difficult for them to keep up. So in addition to the raw challenges of actually just storing and processing that amount of data, there are some real issues with this scale of genomic data generation. One of those is it's actually quite hard to just move this amount of data from one place to another. 10 petabytes of data will exceed the bandwidth of basically any network connection between two places. And in fact, for sufficiently large amounts of data, it is often the case that the most efficient way of moving data from one place to another is to download it all onto individual hard drives, load those up into a truck, drive the truck to another place, and then download it at the other end, which is completely ridiculous in the internet era. This is becoming somewhat easier in the era of the cloud, where we can just store data in one centralized place, but simple storage and movement logistics are still hard. A real non-technical challenge to analyzing the scale of data is the fact that consent has been collected over many decades. And what that means is that people who participate in these large-scale genomic studies were often consented at a time when genomics didn't exist and where we didn't really think carefully about the prospect of global data sharing of data, so that makes our lives much more complicated. There are objections to data sharing by the people who generate it. So many industry-funded collaborations are not interested in making their data available to the world because they see it as something that will benefit their corporation by keeping it secret. Some of you may be shocked to hear that there are even some academics that don't believe in sharing data. And so all of this makes our life a little bit harder as well. But then one of the big challenges that we've had to overcome in our work is the fact that every single person who sequences genomes tends to use their own slightly idiosyncratic pipeline for processing and analyzing and looking at genetic changes in that data. And so that means if we were to naively take the output of each of those different pipelines and just try to munge all of that together into one collection, what we would end up with is a data set where the interesting differences were actually dominated by technical errors rather than by real biology. And so what we need to be able to do is to take as much data as we can get our hands on, process that through exactly the same pipeline, and call genetic variants in exactly the same way across large amounts of data, and then make that data available to the world. And that's what we've been trying to do over the last 4 years. So when I started my lab back in 2012, one of the first projects we worked on was an analysis of exome sequencing data from patients with various rare forms of muscular dystrophy. And one of the things we realized very quickly is that the existing databases of human variation were actually inadequate for analyzing the data we were finding in our patients. What we wanted to do, of course, in the context of a patient, is to take every genetic change that we observed in that person's genome or exome and figure out if that had ever been seen before, if so, how common it was in the human population, and if any of the other people who carry that particular variant also have the same disease. And so the two data sets that we had available for asking and answering those kinds of questions at the time were the 1,000 Genomes Project, which consisted, despite its name, consisted of about 2,500 genomes from people of diverse ancestry. We also had the NHLBI's Exome Sequencing Project, or ESP, which contained about 6,500 individuals of either European or African American ancestry. And then through a process that took about 18 months, that I'm not going to describe in any detail at all, but was fairly painful, both politically and technically, we were able to release in October 2014 a brand new collection of sequencing data consisting of exomes across nearly 61,000 people. So this data set, which is called the Genome Aggregation Consortium, or EXAC data set, was by far the largest collection of human genetic variation that existed at the time, actually by almost an order of magnitude. And that has been used very heavily over the last couple of years. And then in October last year, we were able to release a new version of this data set. This is now called the Genome Aggregation Database, or Nomad. We like, as Eleanor pointed out last night, we like our summaries of our projects to contain idiosyncratic capitalization and silent letters, just to be confusing. So Nomad contains about twice the number of exomes that are present in EXAC, and also for the first time incorporates a large number of whole human genomes. So that means we can look at both the protein coding bits of the genome as well as the non-coding bits of the genome. And importantly, all of this data was, these data were aggregated, not sequenced by us, but were collected by aggregating them together from many other sequencing studies. I'll talk about those studies in a moment. They were all processed through the same pipeline, and then we called genetic variants jointly across all of those samples simultaneously, and this has required a huge amount of effort to get it done. But the end result has really been worth it. So the reason we built the EXAC database in the first place is because we wanted to improve our ability to filter the genetic changes that we found in rare disease patients, and EXAC is much more powerful than previous databases for doing that. So just to illustrate that, here we have, on the left-hand side, the number of variants that remain in an average person's exome from 5 different continental groups. The different color represents different ancestry groups. for analysis that was done using the ESP project, which contained about 6,500 exomes. And so if you apply a frequency filter of about 0.1%, so that's something you might imagine applying in the context of a rare dominant disease, you end up with up to 1,000 or almost 1,000 candidate variants that remain in a person's exome. If you do exactly the same analysis, but now you're using EXAC, which is 10 times as large and much more diverse, you get an order of magnitude reduction in the number of candidate variants that show up in that person's genome. So EXAC is a much more powerful way of filtering the genetic changes that are found in rare disease patients, which is great because that's one of the key things we built it for. I'm not quite sure what it's... Okay, all right. So it's worth spending a tiny bit of time before I move on to the science we can do with this project, just emphasizing who exactly has ended up in these databases. So again, we didn't sequence these people, we aggregated them. We are data parasites. We're opportunistic collectors of data that was sequenced for other purposes, and what we do is just take that data and reprocess it and put it together in the same project and make it available to people. So the people who have been sequenced in this project mostly consist of people from case control studies of complex adult onset diseases such as type 2 diabetes, neuropsychiatric conditions such as schizophrenia and bipolar, and cardiovascular disease, particularly early onset heart attack. So that's where most of these people come from. We have, as best we can, depleted these collections of people who either have very severe early onset diseases like muscular dystrophy or autism or pediatric cancer, although some of them may well remain. And we've also removed their first degree relatives. And it's worth noting, though, that some severely ill people do actually remain in this data set. So we can't get rid of all of them because we don't have perfect health information. And also, some of the people may not have disease right now, but they will go on to get disease later. And that's important for the project that I'll talk about at the end of this talk. And then finally, although these samples often weren't collected for the purpose of doing large-scale genetic studies across many different phenotypes, for about 2/5 of them, we do actually have the ability to go back and get more clinical or phenotype data. This, in some cases, consists of really deep clinical data. So for some of the people who are in, for instance, the biobanks from Finland in this collection, we can actually go back and look at every diagnosis that's ever been made for these people over the course of the 25 to 50 years of their life. So this slightly hypnotic rotating GIF on the left-hand side here gives you a snapshot. This is actually one of the first analyses we did on the new Nomad dataset. And what this shows you is the ancestry of the individuals who actually came into this collection. So this is using an analysis method called principal component analysis, or PCA. It's very simple. Basically what it does is it takes all the information that's present in the genetic data of these individuals and condenses it down so that we can view it along three dimensions. And that those 3 dimensions are what we're basically rotating around here. And so the nice thing about that is it allows us to cluster these individuals by how closely genetically related they are to one another, which corresponds to their genetic ancestry. So you can see here very clear, although somewhat diffuse clusters corresponding to European ancestry here in orange, and there's these little jut-offs here which correspond to individuals of Finnish ancestry and Ashkenazi Jewish ancestry. It's a big cluster of Latino samples down here. We have South Asian samples up here, African ancestry over here, this little cluster of East Asian individuals over here. So this is absolutely by no means representative of the entire spectrum of human genetic variation. We are missing, for instance, the vast majority of variation that exists in the African population. We only have a couple of populations in here. We're also missing the whole of the Middle East, and importantly for this audience, pretty much all of Oceania. So Australian, New Zealand populations, Polynesian populations are also completely missing here. But what it does mean is that for many of these continental populations, we have often thousands, you can see the numbers over here, thousands of individuals from these continental populations that we can use to look up any variant that we find in a patient of that ancestry who's turned up in a rare disease clinic. We can look up to see how common that variant is in that population. So still a lot of work to do, but we're getting there in terms of collecting information across a large chunk of the human population. Now, our purpose in generating these projects was not just to benefit our own rare disease research, but to make sure that anyone around the world could take advantage of the same data sets. And so pretty much as soon as we had the data set to the point that we were comfortable that it was actually working, we had gone through quality control and it seemed to not be a total disaster, we made that data available through a series of websites. There's exec.broadinstitute.org and more recently nomad.broadinstitute.org. If you go to those browsers, which were built by people in my group, what you can find is information for your favorite gene. Here's one of mine. The blue peaks here represents the coverage, that is the number of reads that we have on average across our 60,000 or 120,000 individuals, for exomes shown here in blue and for genomes shown here in green. And then below that, we have a list of every variant that we've observed across our entire collection of individuals. And you can click on those to learn more information about how common that variant is across different populations and how likely we think it is that it's actually a real variant. So there's a lot more information there. And anyone who's interested in human genetics, of course, can look that up. And there's absolutely no restrictions whatsoever on analysis or publication from the data set that's available here. And you can download all of the information here in VCF form as well. So there's lots of things you can do with this data set. I wanted to just briefly mention one of them, and that is work that's been led by Kaitlyns, a graduate student in Mark Daly's lab, who's been working with my lab for this project. What Kaitlyn was interested in is not just the variants that were actually present in the data set, but the variants that were missing from the data set. And to study those, what she did is to build a statistical model that allows her to predict for any given gene in the genome how many variants of a particular functional class you would expect to see by chance. So that means if the only process that was acting on that gene is random mutation and natural selection had been removed, how many such variants we would expect to see pop up in a collection of 60,000 people. And you can see here on this plot, here we're looking at synonymous variants, which are variants that are silent, that is they don't disrupt the protein coding region of the genome. And on the x-axis, we have the number of variants per gene predicted by Kaitlyn's model. On the y-axis, we have the number that were actually observed in 60,000 people, and you can see the fit is exquisitely good. So in classes of variation where there is no selection, Kaitlyn's model predicts almost perfectly how many variants we should expect to see in each of the genes in the human genome. But as we move towards classes of variation that do affect gene function, and where we expect the natural selection will be acting, that fit becomes much worse. And the reason for that is very simple. So here we're looking at loss of function variants or protein truncating variants, variants that we expect to completely break the genes that they're found in. And you can see here that the vast majority of genes fall below the line. And the reason for that is that for most genes in the genome, loss of function variants are bad. They disrupt a gene that's actually doing something useful in the cell. And as a result, most of those variants tend to get removed from the population by natural selection. So there are fewer of them than we would expect to see by chance. So that's no surprise at all. We're not going to win any Nobel Prizes by proving that natural selection actually exists. But what we can do with 60,000 people in this picture is for the first time calculate how far below the line each gene falls. And that gives us a quantitative estimate of how strongly natural selection is actually acting on that particular class of variation. So just to give you one example, here's a gene called DINK1H1. We know exactly what happens when you break this gene, break a single copy of this gene. De novo missense and loss of function variants in this gene are associated with a whole host of horrible early onset neurodevelopmental phenotypes like intellectual disability, seizures and other phenotypes as well. And what we see in this gene is that Kaitlyn's model predicts almost perfectly how many synonymous variants we would expect to see, but we're missing about 2/3 of the expected missense variation and almost all of the expected loss of function variation. So we expect to see 160 loss of function variants in this gene, and we actually only observe 4. And of the 4 that we observed, 2 of them are artifacts. One is a sequencing error, one of them is an annotation error, and there are 2 people carrying these variants in the exact population who probably actually do have one of these diseases. So nonetheless, we could tell, even without knowing that this was associated with disease, we could tell just from this lack of functional variation in this particular gene that something catastrophic happens when you break this gene, even a single copy of this gene. So that's a case where we know what the disease is. Here's a case where we don't. This is a gene called UBR5, where we know that it falls in the ubiquitin pathway. We know if you knock it out in mice, it kills them during embryogenesis. Very little else is known. But here we see a picture that's very similar to what we see in DINK1H1. So that is a well-calibrated model for silent variants, a substantial reduction in missense variation, that is variants that change amino acid sequence, and a near complete absence of loss of function variation. So this tells us, although we don't know what the disease phenotype is, that there likely is some relatively severe phenotype associated with the loss of one copy of the UBR5 gene. And we can apply this, of course, to any of the genes in the human genome. When we do that, what we find is that there are just over 3,000 genes with a near complete absence of loss of function variants or protein truncating variants in the population. What that tells us is that those genes are almost certainly, again, disease-associated genes. And indeed, that set of genes contains almost all of the known haploinsufficient disease genes, where haploinsufficient here means genes where if you knock out one copy of the gene, it results in a severe disease. So that, as we expect, contains almost all of those, but importantly, almost 3/4 of these genes, we really have no idea what they are. There's no phenotype associated with mutations in those genes. In fact, for most of these genes, we often have absolutely no biochemical information as well. So that's really interesting because the genetics tells us that these are interesting. We don't know exactly what they do, and these are now being heavily prioritized in studies of follow-up studies of a whole range of different diseases. including work that Mark Daly is now leading in autism and schizophrenia, where he has shown that in fact this collection of 3,000 genes contains almost all of the signal for de novo protein truncating variants in autism cases. So there's lots of work ahead for us in Nomad, and I just wanted to finish this out before I go on to two stories that explains how we can use Nomad to learn more about gene biology. So we're working on 2 papers at the moment resulting from coming out of the Nomad work. One of them is on protein truncating variants and their effect on genes across the human genome. A second of them is looking at the depletion of variation in particular chunks of the non-coding regions. So applying the type of constraint model that we've done across genes and now applying that to regulatory regions. We are calling structural variation. Our analysis pipeline to date has really focused on very small types of variation, so single nucleotide variants and indels. And we're now expanding that up to include things like deletions, insertions, and inversions. And we're moving all of our analysis pipeline over onto the Google Cloud so that we can stop being held back by not being able to scale based on the physical hardware that we have currently existing at the Broad. And then finally, our mission this year, by the end of this year, is to apply and complete a variant call set across just over 50,000 human genomes. And so we hope to be able to release publicly by the end of this year a call set that will actually likely be over 60,000 human genomes. So we still have a lot of work to do to get there, but we're pretty confident we will hit that number. Okay, so I wanted to spend the rest of my time, let me just check how I'm going for time. The rest of my time today talking through 2 ways in which we can use the variants that we collect from these large repositories of human genetic variation to understand the ways in which genetic variation can impact gene function. And so the first of those will be the Human Knockout Project. So we'll talk about actually leveraging naturally occurring genetic variation to identify rare individuals who are completely lacking the function of a particular gene, and then using that to learn more about the function of that gene. And then secondly, I wanted to talk through use the example of prion disease to talk through how we can actually leverage these data sets to understand the penetrance, that is the risk that someone who carries a particular genetic variant will actually go on to get the disease, to get a particularly severe rare disease. Okay, so let's start with the Human Knockout Project. So again, going back to this slide, talking about reverse genetics in humans. So again, that is starting with people who we know have disruptive mutations in a particular gene, and then going through using this matrix of human genetic variation to identify those people and then learn more about their phenotype. And again, just a reminder that about 40% of the people in Nomad are actually recontactable. They have a whole variety of information about their phenotype that may be available. So we can use this in theory to learn more about where each of the 20,000 or so genes in the human genome sits along this spectrum of tolerance to loss of function variation. So this spectrum is pretty theoretical. We don't actually know where these boundaries are or where these particular numbers are. But we would expect, if we were to arrange all of the genes in the human genome in some reasonable way, what we would see is that there are some genes over on the left-hand side here where loss of function variation is extremely disruptive to human fitness. So these might be cases, for instance, where knocking out a single copy of the gene results in very severe disease. And on the right-hand side, we have genes which are what we would call non-essential. So these may be cases where complete inactivation of a gene, that is homozygous inactivation of a gene, could actually result in a human being who is perfectly healthy, or at least doesn't have any disease that is severe enough that they would end up in a clinical genetics clinic. And we can use lots of different ways to get at where a gene fits along the spectrum. So we've become extremely good at looking at variants in the middle here. So these are variants where either heterozygous or homozygous inactivation results in a severe disease, either a dominant or a recessive severe disease. And as I mentioned at the beginning of the talk, we found now about 4,000 genes associated with those very rare diseases that mostly fall in this middle part of the spectrum here. Now, we don't know a huge amount about the stuff on the left-hand side here. These are often variant genes where messing with them results in embryonic lethality. And so as a result, we know that roughly 30% of genes in mice, if they're knocked out, result in a loss of the embryos of those mice. We don't know many of those genes in humans, and most of the corresponding genes in humans are actually not associated with disease, suggesting that, again, that messing with them in the human situation results in a loss during embryogenesis, and we just never see the humans who carry those mutations. But we can learn more about those genes by looking at their constraints. So the study the analysis that I showed you earlier, where we look at genes that are actually missing variation in the population, we think points us pretty heavily towards genes that fall on this end of the spectrum. But we also actually don't have a lot of information about genes on the other end of the spectrum. That is the spectrum where messing with those genes results in some kind of phenotype, but often not a phenotype that results in people going to a clinical geneticist. So to study those people, you can imagine looking at loss of function variation in very large numbers of individuals from the general population is the right approach. These are the types of genes where human knockouts may actually be found, and they may be really informative about what those genes actually do. So there are a couple of tricks we can use to enrich for populations that are actually likely to have true knockouts, that is homozygous inactivation of both copies of a particular gene. And these have very different effects on the types of genes that and the types and the prevalence of genes that we're able to see knocked out in these populations. So one of the things we can do is to look at bottlenecked populations. These are populations that have gone through a relatively recent small founder population. So this might be the case, for instance, in Finland, and where that founder population has then expanded up to grow to a very large number of people, so 5 million people in the case of Finland. So these bottleneck populations, because they went through a very small number of founders and then expanded up, often actually carry relatively deleterious mutations at relatively high frequency within the population. And that's something we can leverage to learn more about variants that actually do cause disease, like recessive disease variants, for instance. But we would also expect that we will see some true human knockout genes that come through that particular bottleneck as well. And because of the nature of this bottleneck, what we will find is that relatively few genes are knocked out in these populations, but the genes that are knocked out will be knocked out often in many people. So that gives us more statistical power to look at the exact impact of human knockouts on particular phenotypes. A second approach that we can imagine doing is to look at consanguineous populations. Consanguineous just means populations where there is a high rate of parental relatedness, that is, people who mate together who are related to each other, and this exists typically due to cultural practices that result in high levels of cousin-cousin marriage or uncle-niece marriage often. So in these consanguineous populations, again, we will see a large number of individuals who carry homozygous loss of function variants, but here these will be scattered across many of the genes in the human genome. And the reason for that is that consanguineous marriages basically can take any rare variant that exists within the family and happens to be carried by both of those parents, and then what we would call homozygose that. That is, take that and make it that the offspring of those individuals carries 2 copies of that broken gene. And so the end result will be a large number of genes that we find knocked out, but typically a relatively small number of individuals who are knocked out for any one gene. So we've been taking both of these approaches to look for human knockouts across a number now of different populations. In 2 cases, we've been able to study consanguineous individuals. These are both published now. So we've been working with the East London Genes and Health cohort led by David Van Heel and Richard Trembath. This is a population of mostly Pakistani descent living in the UK. We've now sequenced well over 3,000 individuals from that population. And then secondly, working with Dinesh Salaheen and Sekar Katharisan, we've also studied a set of Pakistani individuals living in Pakistan. We've now sequenced about 10,000 individuals from that population, about a third of whom are consanguineous. So in these 2 studies, we really get a good handle on the distribution of homozygous knockout mutations across these consanguineous populations, which I'll talk about on the next slide. Another very large-scale analysis that we've been pursuing, and this has been funded by a 4-way pharmaceutical company collaboration between Merck, Eisai, Biogen, and Pfizer, is to pull together as much information as we can get about the genetics of the Finnish population. This is work that's been led by Arno Pelotier in Finland and Mark Daly at the Broad. What we now have in Finland is a collection of incredibly well-phenotyped individuals. There's about 500,000 people in a whole variety of different Finnish biobanks, about 400,000 DNA samples that have already been collected. 20,000 of those we have now sequenced, either exomes or genomes, and a much larger number now we have genotyping arrays that have been run. And so that gives us an enormous amount of information about the distribution of even relatively rare variants across the Finnish population that we can collect with this glorious electronic medical record and trait data from those biobanks. And so we've learned a lot from this process already. The first big lesson from the 2 studies that I mentioned in the Pakistani populations is that looking at consanguineous individuals gives us an enormous yield of rare homozygous knockouts. So nearly half of the individuals of consanguineous origin in these populations carry at least 1 rare knockout gene. That's way higher than what we see in a European population, for instance, where that number is closer to 1%. So that's a huge enrichment that we can really use to study the effect of particular knocked out genes on the human organism. A second big lesson is that this Finnish biobank data is extremely powerful if we want to be able to go from genotype through to phenotype. And so really having data that's been collected over many decades on these individuals and often multiple questionnaires collected across many years as well as electronic medical record data combined is extraordinarily useful to build up a much richer picture of exactly what is wrong with a particular individual and across many samples. And so far we've been able to discover over 2,500 genes with homozygous loss of function in at least 1 sample across these 3 populations. So we're starting to get well over 10% of the human genome that contains at least 1 knockout, and there's already been some early insights into human biology. So I'll just give you one example of that. This is a gene called Apo C3. It was well established, we've actually known for over 5 years, that heterozygous, that is single copy loss of function of the ApoC3 gene, results in a reduction in triglyceride levels, that is fat levels in the blood, and also reduces people's risk of heart attacks, early onset heart attacks. But what we weren't able to see from these, even from relatively large-scale population studies, is what happens if you knock out both copies of the ApoC3 gene. And this is really important because we already have 4 different pharmaceutical companies that are developing inhibitors to this gene, and it's really unclear what would actually happen. We know that 50% reduction of the protein product is probably okay, but it's really unclear what would actually happen if you developed extremely efficient inhibitors that completely knock down the function of ApoC3 in a living human. So we set about trying to explore that in the Pakistani population. So what we found, and this is work that's been led by Dinesh, Pradeep, and Saik, is in our initial analysis of 10,000 sequenced individuals from the PROMIS study, that is the Pakistani group in Pakistan, for whom we had access to blood plasma data, we were able to show that these people are in fact complete knockouts for ApoC3, both copies are inactivated, and the ApoC3 protein levels in their serum are down to 0. So there's a complete absence of any protein expressed from this particular gene. So that's useful in that it tells us that ApoC3 knockouts can actually survive to adulthood. But we can learn a lot more about these individuals by studying these tremendously large pedigrees that exist in this particular population. So here's one pedigree. This individual in purple here is an individual who was a knockout for the ApoC3 gene. So zooming in on that person's pedigree, here you can see he's married exactly once, he's had 9 children. And so we then set about genotyping each of the 11 individuals within this family to see whether any of the other children in this family by chance may actually also have ended up as being knockouts for this gene. And what we found, to our surprise and delight, was that the wife is also a homozygous knockout for the ApoC3 gene. So this is one of those cases where, despite the random mating of humans, we've ended up with exactly the mating that we would want in this particular family. And as a result, of course, all of the children are obligate knockouts for the ApoC3 gene as well. So we were able to get our samples then from 7 of the kids, and that gave us 9 individuals in one family that we could use to study the effects of the loss of the complete loss of this protein on various different aspects of biology. And one important outcome from this is that these 2 individuals up here, both of whom are now in their 60s, appeared to be relatively healthy. So there was no major clinical syndrome that appears to result from the total loss of ApoC3. And again, that's extremely reassuring to the pharma companies that are running these analyses. But we can also do intervention studies in these families. So what we did is a very simple dietary fat challenge. This basically meant taking individuals who are knockouts or wild type, that is normal for the ApoC3 gene, and expose them both to a dietary fat challenge, which in non-technical terms meant giving them a huge fatty milkshake. They could drink that, and then we measure the fat levels in their blood over time. And what we see when we do that is this. So individuals who have functional copies of the ApoC3 gene, their fat levels spike over a time course of hours after drinking a milkshake containing lots of fat. People who have ApoC3 knocked out, their fat levels change barely at all. So that tells us that if you're missing this protein, there basically is a fundamental defect in the absorption of fat, and that results in this incredibly low basal triglyceride levels in the serum of these individuals. And that would be expected, if anything, to result in even stronger protection against myocardial infarction. So on the strength of this, multiple pharma companies are now proceeding forward with programs targeting very substantial reductions in ApoC3 levels in adult humans. So there's a lot more that we can learn from these knockout individuals. These plots, which are extrapolated forward from the relatively small numbers of samples we sequenced to date, give us an idea about how many more human knockouts we could get if we were to sequence more, larger and larger numbers of samples. And basically that number grows much more rapidly if we sequence individual populations with high levels of consanguinity like Pakistan, and it obviously grows much more modestly if we were to sequence African or European populations. And these numbers on the x-axis are not completely ridiculous. Here we're talking about 50,000 samples sequenced down here. We're actually getting pretty close to that in the Pakistani population. Between various different collaborators, we're now up at about 40,000 individuals of Pakistani origin who have been sequenced. And we would expect that to approach about 100,000 individuals over the next couple of years. So with 100,000 samples, with 100,000 samples, we will be approaching the point where we have about 1/3 of the genes in the human genome where we've seen at least one homozygous knockout individual that we can study. Now, there's only so much you can learn from one individual, but I think combining that with data from model organisms and other analyses will give us a lot of information about the biology of those genes. So there's still plenty of challenges ahead though. One challenge remains that although we can predict loss of function mutations relatively well, that is, variants that are predicted to break the function of a protein coding gene, for some of those we find out that there are various ways that the cell can actually escape loss of function. And so I think we will need to move towards having large-scale RNA-seq and high-throughput experimental assays to resolve exactly what the functional impact of each of these variants are. Secondly, we still have a lot of work to do to handle the amount of data that's required to sequence these number of people. And then finally, it's important to remember that human genetic variation, although it's something that I love and that we spend a lot of time thinking about, is really only just one line of evidence for understanding biology, and that's no surprise to this crowd. Human knockout is equivalent to lifetime whole body exposure to the loss of a particular gene. That's not always the thing that we want to know when we're studying the function of a particular gene. And unless we integrate that with all the other sources of information that are available, such of course as model organism information, we'll be missing a big chunk of the picture. So human genetics is useful, but it's far from the only thing that we need to be studying. And again, that's not a lesson that won't resonate here. Okay, so with that, I'll move on to the final story that I wanted to tell tonight, which is a story about how we can use these large-scale databases of human variation to learn more about how likely it is that a given variant in a disease gene will actually cause disease in that individual. So this is work on a horrible set of diseases called prion diseases, and it was led by Eric Philab-Minikel, who at the time was an analyst in my group for about 2 years. And Eric had a very personal reason to be interested in studying prion diseases. So this is Eric's wife, Sonya, shown here on the left. She's shown here at the beginning of 2010 with her mother. At that time, at the beginning of the year, Sonya's mother was perfectly healthy and there was no sign of any disease at all. Over the course of 2010, she underwent a terrifying cognitive decline and passed away actually before the end of the year. It was completely unclear what caused this decline. But on autopsy, it was revealed that her brain was full of protein accumulations called prion tangles. And it was clear that she had died of one of a number of different prion diseases, in this case a disease called fatal familial insomnia. So this is in some cases a genetic disease. I'll tell you more about that in a minute. So as a result, Sonya's mum was sequenced for the PRNP gene, which is known to be the gene that contains variants that predispose towards prion disease. And she was found to carry a mutation that had previously been reported as a dominant and basically 100% penetrant cause of severe prion disease. So that meant that Sonya herself had a 50% chance of carrying that particular mutation, and if she did carry that mutation, she would then go on to suffer the same fate as her mother. Eric and Sonya then had to decide whether Sonya would get tested, and they agonized about this for a number of days, and then they decided that in fact they would go ahead and get tested. And in early 2011, they received a report from a clinical geneticist that told them that Sonya does actually carry the D178N mutation, the mutation that killed her mother, and that means that Sonya, who at the time was in her early 30s, was destined to suffer the same fate. So there are many different ways of responding to this kind of information, and Sonya and Eric responded in a way that I think many of us to this day find incredibly inspirational. They quit their jobs. Eric at the time was a town planner. Sonya had graduated from law at Harvard. So both of them ended those careers and they began retraining as biomedical scientists with a single-minded goal towards coming up with a therapy for handling prion diseases in the 20 years that they have left before Sonya suffers the same fate as her mother did. And so at the time that Sonya learned this news, she had, of course, one massive question in her mind, and it was a question that she asked the clinical geneticist. Given that I carry this particular mutation, how likely is it that I will actually go on to manifest this disease? Is this 100% certain, or is it 50%? What is my risk? It's actually very difficult to answer this, and it's difficult to answer this because of this question of penetrance. That is the probability that a carrier of a specific disease genotype will actually go on to get that disease, which turns out to be largely unknown for most of the variants that actually cause rare, severe diseases. For some of them, there's an underlying assumption that they're fully penetrant, and that's true. A lot of the time, we actually don't know. We often don't have large enough data sets to really get a good estimate of that number. And penetrance is known to be incomplete and variable for many disease-causing mutations. Perhaps the most famous example, of course, is the BRCA1 mutations, which cause disease in somewhere between 60 and 80% of the women who carry them. But some women who carry these mutations will never go on to get breast cancer, and it's not really clear why. And again, for most disease-causing variants, we actually have absolutely no idea what the underlying penetrance estimates are. So here's what we know about prion disease. It is a rare disease, although not that rare. Actually, about our lifetime risk of getting prion disease is about 1 in 5,000. And the interesting thing about prion disease is that although 15% of the cases are genetic, Most of the cases, as far as we can tell, are sporadic. That is, there appears to be no specific cause, but at some point during a person's life, prion proteins begin to accumulate, and once they do, there is a positive feedback loop that results in all of the prion protein in a person's brain accumulating, resulting in these horrible prion accumulation diseases. And of course, there is also a notorious 1% of prion diseases that are caused by infections, and that is by the ingestion of meat that contains a particular type of unfolded protein. And if you ingest that and you're unlucky enough to have one of those molecules propagate into your brain, that can then trigger, again, prion accumulation within the brain and death. The most notorious example of this, of course, is the variant Creutzfeldt-Jakob disease epidemic in the UK, which was triggered by the ingestion of beef contaminated from animals who had mad cow disease. So the genetic cases are known to be caused by a whole variety of different dominant gain-of-function mutations in the PRNP gene. And we actually know a lot about these mutations because almost every individual who gets prion disease in industrialized countries gets reported to surveillance centers. So that means that nearly all of the diagnosed cases actually have the PRNP gene sequenced. But again, like most diseases, the penetrance of these variants is very poorly characterized. So the clinician who Sonya asked that question wasn't able to answer the question about how likely it was that she was going to be able to go on and get the disease, except to be told that it was very likely. So Eric, with help from my team, then set about answering that question in a much more rigorous and robust fashion. What he did was he firstly set out with I think, perfectly reasonable assumption that the variants that cause prion disease in aggregate should be no more common than the disease itself. So that is, because this is a dominant disease, it must be the case that all of the variants combined must have an aggregate frequency that is less than the disease itself. That's a reasonable and important calculation that will come up in a second. So what we were then able to do was to collect a whole bunch of individuals with information on the prion gene from the general population. At the time, our 61,000 individuals from EXAC, some of whom may well go on to get prion disease, but none of these were ascertained on the basis of neurodegenerative diseases. So they should be broadly representative of the general population. We're also, through collaboration with 23andMe, able to get access to about just over 500,000 customers from this company, all of whom had consented to have their data used in research, and they had been genotyped for 16 variants that had been reported previously to be pathogenic, that is, causes of very severe prion disease. And then, so that gives us our controls. In terms of collecting cases, Eric, through a pretty remarkable series of feats of political jiu-jitsu, was able to collect 10,500 individuals who had prion disease and had the PRNP gene sequenced. And this actually amazingly represents basically all definite or probable prion disease cases in the US, Europe, Australia, and Japan over 15 to 20 years. This is basically as large a collection of prion disease cases as we are ever likely to get. And this collection allowed us to really start to compare the frequency of the variants that we found in our cases to the frequency and controls to learn more about how likely each of those variants is to cause disease. So we can estimate with some hand waving how many people in our EXAC collection of 61,000 people will actually go on to get prion disease due to one of these genetic mutations. And that number, I'm not going to go through the calculations here, they're relatively robust. That adds up to about somewhere between 1 and 2 individuals within our 61,000 people that will actually go on and get this disease. But when we looked at the sequencing data from the PRNP gene across our 61,000 samples, what we found is a number that's much higher. So there were 13 different variants that actually were previously reported to cause prion disease, and the total number of carriers of all of these variants combined was 52. That is 30 times higher than what we would expect to see given the rate of prion disease in the population. So there's a couple of possible explanations here. It could be that prion disease is terrifyingly higher than what we thought it was, but that's pretty unlikely given how good the surveillance centers are. It's possible that some of these variants actually don't cause prion disease at all. In fact, it's likely that some of them don't. That is, they were variants that just happened to be observed in people who have sporadic prion disease, and as a result, they've ended up in these databases of disease-causing mutations, but they don't actually cause the disease. And then a final possibility is that some of these variants may actually have some effect on the risk of prion disease, but not 100%. So that is, they may have incomplete penetrance, but we have no idea exactly what the numbers are of each of those variants. So here is how we figured that out. This plot is very simple. On the x-axis, we have the number of times that we have, so for each of the previously reported variants that have been reported to cause prion disease. On the x-axis, we have the number of times that was seen in our 10,500 cases. On the y-axis, we have the number of times that was seen in our 61,000 population controls. And we can use this to filter these variants out into 3 broad categories. On the x-axis, we have variants that are only seen in cases and never seen in controls. And these are variants where we can be actually statistically almost completely confident that these cause disease in everyone who carries them. And unfortunately, that turns out to include Sonya's variant, D178N. So as far as we can tell, no human being who has ever carried the D178N mutation and lived into their 50s has not gone on to pass away from prion disease. On the y-axis, we have variants that are seen at a relatively low frequency in cases, but at roughly the same frequency in controls. And so that what these tell us is that these variants, they're not enriched in the case population, and they almost certainly are completely incidental. There's absolutely no association with prion disease. We can actually discard these. These are errors in the database. That actually fits with the literature on each of these variants. Most of them actually have incredibly unconvincing associations with disease and no links to family history, for instance. And then in the middle, we have this intriguing class of variation, 3 variants that are too common in cases to be completely benign, too common in controls to be completely pathogenic. So these must be variants that have some effect on prion disease risk, but not 100%. And in fact, we can actually calculate exactly what sort of effect those variants have. And that ranges between 0.1%, so that is 1 in 1,000 lifetime risk up here, and down to about 10% risk for individuals who carry that variant down here. So for these 3 variants, although there was some literature suggesting that there might be an incomplete effect on risk of disease, this is the first time we've been able to actually put numbers on a person's risk, given that they carry that particular variant, and that of course has really profound consequences for genetic counselling for the carriers of these particular variants. And just to give you one example about how this information can actually have a real impact on people's lives, we published this paper back in early 2016. It was accompanied at the time by an opinion piece by Robert Green and colleagues. And Robert, who had actually had access to data from our study earlier than the rest of the population, had actually been counselling a person in his clinic, a woman whose mother had died from fatal familial insomnia, where the woman had also found out that she carries the E196A variant, which had previously been reported to be prion disease causing. And she had actually been told, based on carrying that variant, that she was definitively going to go on and actually suffer from prion disease. In fact, E196A is the variant about which we have by far the most statistical confidence that it has absolutely no association with prion disease. And that is consistent with all of the evidence. There's no Mendelian segregation data in the literature. There's no family history in patients with the variant. And the frequency is absolutely no higher in prion disease cases than it is in ancestry matched controls. So we can be very confident this variant actually doesn't increase her risk of prion disease above baseline. And as a result, Robert was able to go back and tell her that she doesn't in fact have a death sentence. In fact, she has no more risk of prion disease than anyone else in the population, which of course is completely transformative. So for Eric and Sonya, the news was not great from this study. What we learned from this is that, as far as we can tell, Sonya's probability of going on to get this disease, assuming no therapy is developed, is 100%. But there was a little bit of good news, and that came in the form of these 3 variants up here. These 3 variants are variants that are protein truncating variants in the PRNP gene. These are variants that are predicted to completely obliterate the expression of the prion protein. And all 3 of these were found in healthy individuals who survived to quite an old age. Actually, one of these individuals was 85 years old. So these variants, which fall very early in the protein, told us that if you knock out 50% of the prion protein in a person's brain, we actually did Western blots that I'm not showing here to show you that to demonstrate that protein levels were reduced, that's actually compatible with life. And that's also consistent with animal models that tell us that if you knock out the prion protein, it actually doesn't affect human lifespan. And that's important because what it tells us is that if we were to design inhibitors that reduce the levels of prion protein in Sonya's brain, that will actually reduce her risk of prion disease, but likely won't have any severe adverse consequences. So again, it's likely that inhibition of the prion proteins is actually safe. So Eric and Sonya are now both PhD students at Harvard. Actually, quite remarkably, as a result of donors at the Broad, they've been given their own lab at the Broad Institute. They have a fully kitted out biosafety lab that allows them to study prion diseases at the biosafety that's required. They now spend all of their time working on developing therapies for prion disease, now working with 6 different pharmaceutical companies to do so. Sonya is actually pregnant now. She had prenatal genetic testing, and she is due in July with a child who will not carry the prion disease mutation. And so, of course, we wish them extremely well in their venture forward into basically a 20-year process during which they need to be able to figure out how they can actually design a way of knocking down the prion protein in Sonya's brain, getting that through the arcane processes that are involved in regulating the delivery of a particular drug, and then deliver that to Sonya in a way that will actually reduce her risk of going on to get this disease. So in terms of whether or not this will actually mean that Sonya goes on to get the disease, the statistics is relatively clear. Everyone, as far as we can tell, who has ever carried this particular mutation and survived into their 50s has gone on to die from prion disease. But if you ask Sonya this question, she says no. And the reason for that is that she firmly believes that it is actually possible, given how much we know about the biology of this protein and exactly what is required to engage this target. She thinks there is still a very good shot of actually developing a therapy that will save her in the 20 years that she has left. And to be honest, I kind of believe her. So, ending on that note, this is always actually very difficult to transition from that story into anything else, but I wanted to finish by concluding with a couple of points about things we've learned from studying large amounts of human genetic variation data. The first is that naturally occurring human genetic variation is all over the place. Our population is full of it, and we can use it to learn more about the function of individual human genes once we collect enough information about sequence and phenotype data. The second is that the more humans we have and the more diverse their ancestry, the more powerful our resources become. And so I think it is not just the case that we need to sequence more individuals, we also need to think very carefully about how we can sequence more individuals in a way that benefits more populations around the world, and that ensures that rare disease patients, regardless of their ancestry, have well-matched reference samples. And that is something I think that the community, the human genetics community as a whole, has a moral obligation to work towards. And honestly, at the moment, we still don't do a great job of this. Nonetheless, over the next 5 years, it is unambiguously the case that we will collect genome and clinical data for millions of people. And if we can combine that in the right way, and if we can ensure that that data is collected ethically and shared in a way that benefits the rest of the community, this will have a profound impact on our understanding of human genes. If we can combine this with other resources, such as model organism data, cell models, and other information, this really will result in profound new insights into human biology, and also allow us for the first time, I think, to really understand the impact and the penetrance of human disease-causing genetic variation. So this is a tremendously exciting time to be doing human genetics, and to take that human genetics and convert it into an understanding of human biology. So with that, I'll finish by thanking everyone who's been involved in this work, particularly members of my lab. I want to particularly highlight Monkol Lek here. Monkol is a postdoc who actually led all of the EXAC work. Monkol is actually just in the process of starting up a new lab in Yale. He'll be starting that in November. So if there are any graduate students in the audience who are interested in doing postdocs in human genetics, Monkol is open for business. I also wanted to thank all of the members of the production team at the Broad who are responsible for building the infrastructure and for running a lot of the analyses that are required to make this process work. Ben, Conrad, and Matt built the EXAC and Nomad websites. A big team was involved in actually getting the ethics together to make sure that we had the consent to do all the things that we need to do across 140,000 people. The Broad Genomics platform generated 90% of the sequencing data and funded actually a lot of the work that was required for EXAC and Nomad to exist. And Hail is a key piece of software that allowed us to run quality control across 140,000 people simultaneously. And finally, big thanks to all of the 107 now, this is a large number, all of the 107 now principal investigators involved in the Nomad consortium who agreed to donate their data, not just willingly, but enthusiastically to the Nomad project for the purpose of benefiting not themselves, but the rest of the community, and particularly rare disease patients. And thank you all so much for listening. Thanks.
Thank you very much, Daniel, for a superb talk. There must be some questions, I'm sure. Yeah. You get a mic.
All right, so my question is based on the EXAC and Nomad databases. You also have things at Broad like the GTEx and other databases. Is there any attempt to, the genome's only one part of the picture. Is there any attempt to integrate these things so that if I type in a certain variant, if I typed in one of those prion variants, I would see not only from Nomad, but I'd see from GTEx the expression and different things like that?
Yeah, that's a great point. So we're actually in the middle of building a database called DBLOF, a database of loss of function variants. The goal for that database is to take a particularly deep dive into variants predicted to cause protein truncation. And that will actually incorporate not just data from EXAC and Nomad, but also data from the GTEx dataset, so that if we're lucky enough that one of the 900 or so people in GTEx actually carries that variant, you'll be able to immediately see what effect that actually has on allele-specific expression, as well as any deviation away from normal splicing occurred that is triggered by that particular variant. We still have a lot of work to do though. We still only have in GTEx only 900 individuals for whom we have transcriptome data. And I think it's in order to have a much wider sense about how particularly very rare variants impact the transcriptome, those numbers need to get much, much larger. So multi-tissue transcriptome data from many, many individuals will be needed to really start to dig into the functional impact of most of the variants in the genome.
Question, yeah. Martin.
Hi, thanks for a great talk. I'm just wondering what the ETA on the copy number variation data would be. Are you just doing it on the whole genomes or not trying to do it on exomes?
We will do it on both. There's a new copy number variant caller for exomes that's actually just been rolled out in GATK. And that we will be running that across all 126,000 exomes that'll be available. But most of the work, as you guessed, is being done in the whole genome data. So that's 4 different structural variant callers that we run across beginning with our 15,000 currently available samples and eventually scaling that up to all 50 to 60,000 in the next release. We're hoping to have an SV call set released by the end of this year, at least for the 15K. For the big data set, that will likely be sometime in the first half of next year. Yeah.
Okay. Behind you there.
Hi, Daniel. With your consanguineous families, that I always have trouble saying, you're looking at a particular loss of function variant and associating that with phenotype. But I'm just wondering how many other loss of function variants there are in general in those families.
Yeah, it's a great question, because obviously, given that on average you have like a sixth of the genome that is autozygous in these individuals, it's pretty hard to say if you've got one individual, it's pretty hard to say whether a given phenotype is due to that specific homozygous variant or there may just be some other recessive variant elsewhere in the genome. So the average number of rare homozygous loss of function variants in the carrier individuals is 1. So that's ended up being quite useful. 42% of people have at least 1, and the modal number is exactly 1. But nonetheless, we find individuals who have many more rare homozygous loss of function variants. The record so far is 8. One individual who has 8 separate genes knocked out, which is pretty impressive, actually. And so in those individuals, obviously, it's very difficult to make any specific phenotypic correlation with the genotype. And it will require, I think, typically having substantial numbers of knockout individuals, preferably across multiple families, to account for genetic background, to really be definitive in saying this gene, when you knock it out, does this exact thing. For the cases where we only have one individual knocked out, I think that can give us some clues. And if it also lines up with things that we see in knockout mice or other systems, then we might start to believe that that phenotype is actually real. But it's going to take a lot more convincing based on N of 1 to actually get to that level of biological understanding.
Good. Yeah.
For individuals who you've done either whole genome or exome sequencing on, is ancestry self-reported? And if so, how does ancestry or ethnicity correlate with the genomic variation? Yes,
it's a good question. And in practice, we actually ignore self-reported ancestry entirely. So we do have it, I think, in about a third of the samples in the database. But we completely ignore it because the ancestry estimates that we get from our genetic data are now so much cleaner than anything we could get from self-reported data. That we have tended to not look at it too much. In the cases where we have taken a deeper dive, I guess the one place where we have looked a little bit more closely is in individuals who self-report as being African American. And there the estimates are pretty good. Basically, people who report as being African American are 80% African on average, consistent with other studies. But we get a pretty wide spectrum of variation within that. And we find some individuals who self-report as African American who, as far as we can tell, are completely European. So there's a bit of a mix, yeah.
Okay. I'm just wondering about the that curve, the asymptote towards 8,000 genes that you're going to register. That's over a third of all human genes. Have you discovered any incongruence between, say, mouse knockout phenotypes and what you're observing in those individuals?
Yeah, we have. And I'm still not quite sure what to make of them. So in actually both the papers that we put out over the last couple of years on human knockouts in consanguineous individuals, there are genes that we report in those papers as being knocked out where there is a homozygous knockout phenotype that is extremely severe, actually. There's multiple cases like this. So that could be one of two things. In some cases, it could well be that the human biology is different from the mouse biology. That's not a terrible surprise. That obviously happens sometimes. It could be the case that there's a zygosity difference in the impact between humans and mice, perhaps. It may be that for some of those variants, we've just misannotated the loss of function variant. So they do look like we have filtered out the annotation errors as best we can, but it may still be that these humans actually carry a variant that affects a transcript that the cell doesn't need for some reason, or there's some other way that the cell can actually escape the loss of function. That's possible as well. Maybe the mouse phenotype report is wrong as well, but we don't really know. we are very interested in digging into those. Actually, we've got a graduate student at the moment who's done some digging at the comparison between the discrepancies in human and mouse knockout phenotypes. And the plan is to dig into a bunch of those and see if we can actually try to untangle the possible explanations a little bit further.
Is there a question here somewhere?
Oh,
Yeah.
hey.
Just wondering, are you planning to release the clinical data from the patients that are in EXAC or Nomad?
Right, yeah. No, we'd love to. And I think the challenge there's a couple of challenges there. One is that the clinical data in many ways is much more identifying than the data that we have in Nomad. So it's difficult to figure out a way of releasing that in a way that doesn't allow you to triangulate back to kind of individual level genetic data, which we have to be very careful about. So, I mean, as you know, EXAC and Nomad at the moment, we release frequencies across all of the individuals, but we don't release the individual genetic variation calls, and that makes it kind of difficult to combine the two. What I think we will be able to do is for individual variants, we'll be able to hopefully be able to release at least some phenotypic annotation so that we can say, particularly for the loss of function variants, these are variants where carriers have been observed to have this particular range of phenotypes, but not give you detailed per individual phenotype data across those individuals. And so that is the current plan. We've been rolling that out within the consortium, and we're hoping we will be able to release at least some version of that next year.
One last question. We've worked with Daniel pretty hard.
So just following from the last question, with the Nomad and EXAC, the clinical data that you have, the relative ratios of clinical data that you have across the different population groups, how does that compare?
Pretty uneven. Actually, that's a good question. I'm just trying to go through it in my head. So we have a lot of, there's some weird clustering actually within the Nomad dataset. So for instance, if you look within our European cluster, a lot of our Europeans are of Scandinavian ancestry. We have this enormous overrepresentation of Finns, for instance, which is a consequence of the fact that so much human genetics work has been done in those populations. Actually, if you are of Finnish ancestry, your odds ratio for being an EXAC is 70. So there's this huge overrepresentation of those individuals. And for the Finns, of course, the amount of phenotype data that we have is astronomical. So basically everyone in that collection has very deep phenotype data. For the individuals of South Asian ancestry, most of those actually fall into the Promus cohort, our Pakistani cohort. There we have lots of phenotype data, but it's very cardiovascular focused. And then it sort of dwindles down a little bit after that. So that there's a bunch of other cohorts within the data set with longitudinal health information. I think they're thinking through it relatively scattered across the different ancestry groups. And the one ancestry group where we have almost no phenotype data is East Asians, where for various reasons, the cohorts that fall over there, we just don't have good electronic medical record data for. So I don't know, that's a bit of a vague answer. We have, basically, there's no predictable patterns between ancestry and electronic medical record data. And we still have a lot of work to do in particular ancestral groups to really pull that data set together.
Okay, well, look, I think I might draw it to a close there. We've had a real treat tonight. Thank you so much, Daniel, for a superb talk.